Bismillahirrahmanirrahim. I am Dr. Jamil from the Physiology Department, and today's topic is the from the organelles, and one of the very important organelles is your nucleus. Different organelles which are going to study. One of them is the nucleus, and in the nucleus, a very important structure is your DNA. Although most of the part of the DNA you will study in the biochemistry classes, but we have to touch this DNA. And today's lecture or today's topic is the DNA structure or the introduction with the DNA. DNA, you know the nucleic acids, and these nucleic acids are required for what? They are required for the storage and the expression of genetic information. There are two types of the nucleic acids, DNA and the RNA, deoxyribonucleic acid and ribonucleic acid. And then DNA is present in the chromosomes, the mitochondria, and the chloroplast of the plants. And usually this is your MCQ that your DNA is present in, and options are the chromosomes is not there. Option is the mitochondria is there. You have to take this mitochondria, but the mitochondria also contain the DNA in it. Now the DNA, which contains your master plan of life, the events of your life, your type of the face, your behavior, your diseases you have to face in your life, or you can say your age, all those things is stored, or your master plan is present in your DNA, and it contains the genetic code for whole your life. This DNA actually, when it wants to transmit something to the next cell, it has to convert into the RNA. And this RNA, then ribose nucleic acid, is going to form the different types of proteins. These proteins may be the structural one, it may be the functional one, and this is known as the central dogma of the biology. Now, we are going to focus upon the DNA structure. As you know from the previous classes, the DNA is a linear polymer of the nucleotide composed of three things. That means there are three ingredients in your DNA. If you want to synthesize a nucleic acid, you need at least three things to form it. One of them is the nitrogenous bases, and these nitrogenous bases, they are 4 in number for the DNA, and these are known as the adenine, guanine. These are the double ring structures, while we have got single ring structures where they are known the cytosine and thymines. These are the single structures. Structure. Actually, these are the purines, adenine, guanine having the double ring, and we will see in the next slides the structure. Then one pentose sugar joins the third carbon of the first sugar and the fifth carbon of the next through phosphodiesterase bond using a phosphate from the ATP. When you see the diagram, you will come to understand this whole line. One pentose means 5. One pentose sugar joins the third carbon of the first sugar and the fifth carbon of the next through phosphodiesterase bond using the phosphate from ATP. Then we have got the nitrogenous bases which join to the these nitrogenous bases join to the nitrogenous bases in the anti-parallel way. That anti-parallel way means they are not the parallel. So if the one road, suppose you are going to the motorway, that motorway, when you are moving towards Islamabad from Lahore, you are using your direction Islamabad, but the traffic which is coming from Islamabad to Lahore, that is, although these are parallel, but you are moving on one side and traffic coming from the other side. This means the anti-parallel way. Through the double hydrogen bond in adenine between adenine and thymine, and we have got the triple hydrogen bond between the guanine and cytosine with a fixed rule. That is very important. One important point is they are the anti-parallel. Second is there is a fixed rule. That means the abbreviation which you will use at a government college. Although we should not use these things in the lectures, but just to make you memorize the things. Adenine always join to the thymine, and the guanine always going to join the cytosine. As written here, the adenine with the thymine and the guanine with the cytosine. This is a fixed rule that adenine always going to join the thymine, while guanine is going to join the cytosine. Then ribose. What is the difference between the ribose sugar and the deoxyribose? Because the deoxyribose, the DNA ribose is the RNA. Here you see in the red one, that ribose has got both the hydroxyl groups over here. The third carbon, the second carbon, both of them they have got the hydroxyl group. If we remove this oxygen from this hydroxyl, then this will be known as the deoxyribose. So this is the difference between the ribose and the deoxyribose. Just the removal of one oxygen from this hydroxyl group, we are going to form a deoxyribose. Here is written the ribose have hydroxyl at the position number 2. If we remove one oxygen from the position number 2, ribose will be converted into the deoxyribose. Then this is the basic structure of the nucleotide. That means it contains three things. One of them is the phosphate, sugar, and the nitrogen bases. So this nitrogen base and sugar, and this is joining of this phosphate group with them, this is known as the nucleotide. Here the nitrogen bases of DNA, there are different structures. These are pyrimidines and these are the purines. Pyrimidines and purine, nitrogen base of DNA. You can see the biochemistry books, their structure in physiology. You are not supposed to memorize the chemical formulas of those things. These are the requirements of the biochemistry classes. Now, there are total 5 nitrogenous bases, and here in this diagram you will see some are found in both DNA and RNA, DNA and RNA, DNA and RNA, but 2 of them found only in the DNA, 1 of them is found only in the DNA, 1 is found only in the RNA. In the RNA, there is only one, the uracil, which is found in the RNA. This one. And this is thymine, which is found in the DNA. So, is that cytosine, this adenine, these are double ring structures, guanine and adenine, they are the double ring structures, while this is a single ring structure. The pyrimidines, they have got single structures, purines, they have got the double ring structures. Now, what is meant by the nucleoside and nucleotide? Here you in the diagram, concentrate on this diagram. Here the nucleoside. Nucleoside made up of only 2 things. That is the nitrogenous base, the dark blue, and the green, a sugar ball. So if these 2 are present, this is known as the nucleoside. The base, that is nitrogenous base, along with the sugar, the nucleoside. But if we add tightly a phosphate bond, I have used the word tightly deliberately because just to make you understand the TI, that is nucleotide. If we add a phosphate group tightly, then we are going to get a nucleotide from the nucleoside. So nucleoside is made up of only the 2 things, while nucleotide is made up of 3 things. One is the nitrogen bases, then the sugar, and then we have got the phosphate group. This is again a chemical structure. Here you see there is a triphosphate group, there is a triphosphate, and here we have got the pentose sugar, and these are the nitrogenous bases different present here. So this was a brief introduction about the DNA. In the next lecture we are going to see in detail the replication of the DNA and the repair of the DNA, all these things. Thank you very much.